Hey everyone, and welcome back to ByHollyG. Welcome to today's video. So this one is going to be all about bacteria, the biology of bacteria, and I am planning some videos where I talk about like antibiotic resistance and the gut microbiome and the microbiota. So I thought it would be useful to film this video first so you at least know what bacteria are, what their structure is, just a bit about bacteria. It's going to be a crash course on the biology of bacteria, and if you are looking for a video that you can use to kind of like help alongside this one, I recommend the tour of an animal cell, because you will then be able to compare a bacterial cell with an animal cell. So I hope you enjoy this video. Anyway, as usual, definitely give it a thumbs up if you do, comment down below, subscribe if you are new, and hit the bell so you know when I upload. And yeah, we will dive straight into the video. The first question then is, what are bacteria? So we need to firstly get to know what they are, and bacteria are what we call microorganisms, or microbes, the shortened version, meaning that they are microscopic. We cannot see them with the naked eye, and we have to visualize them under the microscope. They are single-celled microorganisms, but sometimes they will combine temporarily or permanently to form these colonies. Bacteria are also everywhere, like on our bodies, in our bodies. They can live in really extreme environments where most other organisms don't live. They can therefore be extremophiles or extramophiles, for example living in volcanic craters, so really hot and harsh environments. And what's really quite important here is that we have three domains of life. We have bacteria, archaea, and eukaryotes. And the bacteria and the archaea, they are what we call prokaryotes, in contrast to eukaryotes, which includes animals, so ourselves, plants, fungi, and others. And the defining difference between a prokaryote and a eukaryote is that prokaryotes, so bacteria, they do not have a membrane-bound nucleus. So their genetic material, their DNA, is not enclosed in a membrane, because in eukaryotes our DNA is found in the nucleus, and the nucleus is a membrane-bound organelle. And an extension of that is in eukaryotes we also have other membrane-bound organelles. So alongside the nucleus we have like the Golgi, the endoplasmic reticulum, we also have the mitochondria. A lot of the organelles in eukaryotes are therefore membrane-bound, and you'll know that if you watch the video looking at an animal cell in detail. There are other differences between prokaryotes and eukaryotes, and I will touch on some of them a bit later. One obvious one is that bacterial cells and prokaryotes are smaller than eukaryotic cells, but that is just the defining difference that is really important to know, is the presence or absence of a membrane-bound nucleus determines whether it's a prokaryote or a eukaryote. Question number 2 is, what is the genetic material of bacteria? And as I've already said, they have DNA, but it's not found in a nucleus. So bacterial cells, they have a main chromosome, and that is a large circular chromosome that carries essential genes, so genes that are needed for the survival and the reproduction of those bacterial cells. If you took away those genes and that main circular chromosome, the bacterial cell would not be able to survive. And because that's not membrane bound, we say that it is found in a space in the cell, in the bacterial cell, called the nucleoid. And sometimes the term nucleoid will be used to define the chromosome itself, so the DNA, in complex with DNA binding proteins. The term nucleoid is just used in different ways. But additionally, on top of that, as well as the main circular chromosome in bacterial cells, we also have these things called plasmids. And plasmids are small circular molecules of DNA. They are kind of extra chromosomal molecules of DNA because they are separate and distinct from that main bacterial chromosome. And they're really interesting because they are mobile between bacterial cells, which I will talk a bit about later, but they carry non-essential genes. So if you took away the plasmids, the bacterial cell would still be able to survive, because what they provide is kind of like additional or accessory genes. We can say that it's an accessory part of the bacterial cell genome because it allows bacterial cells to, for example, survive in the presence of antibiotics. It carries antibiotic resistance genes, for example. So the next question is, what is the structure of a bacterial cell? And I'm going to show you this all on a diagram. So the first thing to say is that the bacterial cell's genome is basically referring to the main circular chromosome and those plasmids, as we talked about. So that is the entirety of the genetic information in a bacterial cell. As I said, bacterial cells will not have any membrane-bound organelles, and so they won't have any internal membranes, but they do still have a cell surface membrane that surrounds the whole entire cell. But an organelle that isn't membrane bound and is still therefore found in bacterial cells are the ribosomes. And ribosomes are structures that are needed for protein synthesis. They carry out the translation part of protein synthesis, where mRNA is translated into a polypeptide chain. And in bacterial cells, they are smaller than the ribosomes in eukaryotes. So this is all based on density, but in prokaryotes and bacterial cells, we have 70S ribosomes, and in eukaryotic cells, we have 80S. But these ribosomes, they still have two subunits, but the subunit sizes are different again in prokaryotes and eukaryotes. So bacterial cells, they have 70S ribosomes, they're smaller than the 80S ones in eukaryotes, and they have a large subunit, which is 50S, and a small subunit, which is 30S. Whilst the equivalents in eukaryotic cells are 60S and 40S. So the large one is 60S, the smaller one is 40S. I know those numbers don't add up, as in like 30 plus 50 is not 70 and 60 plus 40 is also not 80, but that is due to the way in which we measure the density of the ribosomes using what we call the Svedberg unit. If you're interested, you can read more about that. It's not too important at all, but the important thing to remember is that prokaryotes have ribosomes, but they are just smaller than eukaryotic ribosomes. Now we're just going to look at what is surrounding the cell surface membrane of a bacterial cell. So they have a cell wall, and that bacterial cell wall is composed of a very complex molecule, or a polymer, that we call peptidoglycan or peptidoglycan, however you want to say it. I think it's peptidoglycan, but I say things very strangely, and I've always said peptidoglycan. Because it's a polymer, it is composed of repeating monomer units, and those monomer units are composed of modified sugars, so amino sugars, and a polypeptide, pentapeptide. And beyond the cell wall then, some bacterial cells will have a slime layer or a capsule. A capsule is just like thicker than a slime layer. They are very poorly defined, but it's composed of this kind of like sticky substance that's made of like proteins or polypeptides or both. And the final thing to mention about bacterial cells and their structure is the surface structures. So there are three that we're going to talk about, and the first are flagella, and that is plural, the singular is flagellum. The flagellum is basically used for locomotion, so for movement in most bacterial cells that do move, and the flagella can rotate either clockwise or anti-clockwise, and that determines the direction of their movement. So when it rotates in a counterclockwise or anti-clockwise direction, the bacterial cell moves forwards, and then when it rotates in the opposite direction, so clockwise, the bacterial cell basically like tumbles, and that means that when the flagellum starts to rotate counterclockwise again, it will move in a different direction because it's kind of like been tumbling in space. I just think that fact is so fun about bacterial cells, the fact that they just like swim and then they tumble and then they move in a different direction. The second surface structure then is what we call a pili. Pili are basically these kind of like hair-like structures, and they are used in a process called conjugation, which allows the movement of genetic material between bacterial cells. The final structure, the third surface structure of bacterial cells, are the fimbriae. Again, I could be saying all of this wrong because I never normally say this out loud. They are basically attachment pili. They're shorter than the conjugative pili, and they help a bacterial cell attach to other bacterial cells or like some sort of surface in their environment. The next question is basically, how do bacterial cells divide? And they undergo a process called binary fission, which basically describes the splitting of a bacterial cell in two. It sounds like quite a simple process. It's not actually that simple, you know, nothing in biology is ever simple, but binary fission describes the splitting of a bacterial cell in two, so it has made two new daughter bacterial cells, and that is how it undergoes cell division, and it makes more copies of itself, so the population can therefore grow. In terms of like bacterial growth curves, we have a lag phase, a log phase, and a stationary phase. In that lag phase, the bacterial cell population is not growing too much, the growth rate is very low, and that's because the bacterial cells are just like adapting to a new environment. And then once they have adapted to a new environment, they will grow rapidly, they will show exponential growth, and that is the log phase, and that is when we see their rate of growth being at its maximum, and then they will reach a plateau, so they come to the stationary phase. That is when the population of the bacterial cells remains constant. The rate of cell division and the production of new cells is equal to the death rate, so we just see no change in population size. We then do have a death phase, so when the death rate exceeds the rate of cell division, and that usually happens because there aren't any nutrients left in the media that the bacterial cells are growing in, or the environment, or there's been an accumulation of a toxic kind of like waste product that kills off the bacterial cells. The next question then is, how do we classify bacterial cells? Because there are different ways we can do this, and I'm going to talk about three of the key ways that we classify bacterial cells. The first one is fairly straightforward. It's just based on the shape of a bacterial cell. So those that are completely spherical, they are leucocci, and then we have bacillus shapes, we have Vibrio species, we also have those that are kind of like spirals, they're the spirochetes, and there are other shapes. The second way of classifying bacterial cells then is based on their oxygen requirements. So we have three types. The first are the obligate aerobes. They have to live in the presence of oxygen. They absolutely require oxygen for their survival. In contrast to that, we have obligate anaerobes, and these can only survive in an environment without oxygen. Oxygen is basically poisonous to the obligate anaerobes. They have to live in anaerobic conditions, and anaerobic means without oxygen. And then the final type is the facultative anaerobes, and these can survive either in the presence of oxygen or without oxygen. The third way of classifying bacterial cells then is based on gram staining, and this uses a dye staining protocol. We have gram-positive and gram-negative bacterial cells. The gram-positive ones, they are purple, and the gram-negative bacterial cells are pink. Now, I have never found a way to remember this because I've just studied this for so many years now. It's ingrained in me that gram-positive are purple, gram-negative are pink, but you need to find a way to remember that, because trust me, it will easily slip out of your brain if you only begin to study this stuff. Even if you remember me in this video trying to tell you how to remember it, that might help you. I don't know. Something that might help, that has helped me, it's very strange, but like gram-positive, they are purple, and the O in positive looks like the U in purple. I don't know if that's going to help you. Some bacterial cells, so for example like Mycobacterium tuberculosis, they are neither gram-positive nor gram-negative, and we stain them in different ways. But here we're talking about gram staining, and as I said, it's based on this staining protocol, and it uses crystal violet, which is purple. We then use iodine to like fix the crystal violet, and then we use alcohol to wash out the dye, and then we use a counter stain that's pink called safranin. And the reason why we have gram-positive and gram-negative bacterial cells is all based on the structure of their cell wall. And as we saw previously when we looked at the structure of a bacterial cell, their cell walls are composed of peptidoglycan. In the bacterial cell that I was describing before, in that you have the cell membrane and then the cell wall and then maybe the slime layer or the capsule, that is essentially a gram-positive bacterial cell, because the gram-positive ones, they have a cell membrane and then they have a thick layer of peptidoglycan, and that means that when we stain them with crystal violet and we fix that with the iodine, when we try to wash that out with alcohol, because we have a thick layer of peptidoglycan which basically traps that dye, that purple color, it means that it doesn't wash out, and so they remain purple. Now if we look at gram-negative bacterial cells, they are pink because when we do wash out that crystal violet dye with the alcohol, it can be washed out because they have a very thin layer of peptidoglycan, and so we therefore can only visualize them when we counter stain them with safranin, so they appear pink. Going into a bit more detail then, they have this very thin layer of peptidoglycan, as I said, that's their cell wall, but it is trapped in between two membranes. So the gram-negative species, they have an inner membrane and an outer membrane, and that thin layer of peptidoglycan is found sandwiched in between them. In their outer membrane, they have a molecule that's called LPS, lipopolysaccharide, and that is a really important molecule in bacterial cells. It's specifically found in the outer layer of the outer membrane, because any membrane is a phospholipid bilayer. So it has two layers, and LPS is found in the outer layer of the outer membrane in the gram-negative bacterial cells. The final question of this video is, how do bacterial cells exchange genetic information and genetic material? Because bacterial cells, they show immense genetic diversity, and this genetic information, their DNA, it can be passed down the generations, so from like the parent bacterial cell to the daughter bacterial cells, and that is what we call vertical gene transfer, like down the generations. It's equivalent of me inheriting genes from my two parents, that is vertical gene transfer down the generations vertically. But we also have something called horizontal gene transfer, and this describes the sharing of genetic information between individuals. They can be the same species, they can be different species, but it's not describing the movement down or through generations. And it would be the equivalent of me giving some of my DNA and my genes to you guys watching through the screen. Like, that obviously doesn't happen in humans. It does happen though in bacterial cells alongside vertical gene transfer, and there are 3 key mechanisms or ways in which horizontal gene transfer occurs in bacteria. The first is something we've already mentioned, so conjugation. That is the transfer of genetic material, so DNA, between bacterial cells using those pili, and usually that involves the plasmids being transferred, but we have also seen that whole main bacterial chromosome being transferred through conjugation as well. The second is transformation. Now, transformation basically describes the phenomenon where bacterial cells, they just take up naked DNA from their surrounding environment, and that naked DNA might have come from a bacterial cell that died, for example, and released its DNA, but bacterial cells, they just take up DNA from their surroundings. And the third mechanism of horizontal gene transfer is by transduction, and this basically describes the spread of DNA and genetic information mediated by a virus that we call a bacteriophage. Bacteriophage are basically viruses that infect bacterial cells and attack bacterial cells. They don't attack us. Bacterial cells basically have their own viruses, which is again a pretty cool fact to know. So that is everything about bacterial cells. As I said, it was a crash course on the biology of bacteria. So definitely like this video if you enjoyed it, comment down below, subscribe and hit the bell if you are new, and as always, I will speak to you very soon in another video. Bye!